Yes, students, today's class on malaria in Human Health and Diseases chapter. I'm going to take the malaria, right? So here, malaria, that the meaning of malaria is malaria, it means mal means bad, mal means bad, and area means air. It is spreading from the bad air, right? It is spreading from the bad air. So on those days, the people are believed that malaria means it comes from the bad air. Actually, this is malaria is the caused by the protozoan. From where it comes from? This is from the protozoan. Protozoan disease. That's a pathogen, right? Malaria parasite under comes the where it is from the protozoan, right? The phylum Protozoa, which is that belongs to the protozoan. What do you call? Protozoan, right? So here, that phylum, if you talk about the particular Plasmodium, Plasmodium is the disease causing organism which belongs to the phylum. Its phylum is the protozoa. Subphylum is subphylum is which is the epicomplexa. Epicomplexa, right? Here is the subphylum is the epicomplexa. What is the phylum? Phylum is protozoa. What do you call? Protozoa, right? Subphylum belongs to this is belongs to subphylum epicomplexa. Epicomplexa, right? And class, this is the classification of that protozoan which is the malaria parasite, we call it the Plasmodium. Class is a telosporia. Telosporia. What it's telosporia, right? Genus is the Plasmodium. What it's Plasmodium is the genus. It has a different species are there. Different species are there. One is the Plasmodium vivax, right? And falciparum like that there. Right? We are going to discuss this all in this video, right? So here see what I explained there, the same thing here there, which are the what do you call these Plasmodium. It has a pathogen name is Plasmodium vivax, and moreover the phylum what I discussed there here, this is the epicomplexa is the subphylum. Actually this is the subphylum, right? This is the phylum is the protozoa. Disease which is causes the disease is the malaria. What we discussed here, mala means bad, area means air, bad air, right? It has the Plasmodium vivax or the whatever the species, it can complete their life cycle within the two host. That's why called as the this is called as the digenetic parasite. What do you call? Digenetic parasite. What is digenetic parasite? Right? It means what? No, the quite two host. One is the mosquito. One is the mosquito. Right? Other one is the human. That's why that secondary host is the man, primary host is the female Anopheles mosquito. Female Anopheles mosquito. Why primary mosquito considered as a primary host means here is the Plasmodium undergoes the sexual mode of life. Okay, sexual phase will be happens in the primary host, it's Anopheles mosquito female. Secondary host is the human being. In that, it will be happening the asexual mode. Asexual mode. That's why that is considered as a what you call secondary host, right? Then here is the discovery and what you call that discovery of these all the things. Here is the first one is 1800. 1800. Here is the Patrick Manson. Got it? Patrick Manson, right? He suggested that malaria might be spread by mosquito, not confirmed, but he thought that malaria may spread through the mosquito. Got it? While he is doing or conducting experiments on the filariasis, he found that this malaria might be spread by the mosquito. Who has given? Patrick Manson. In which year? 1800 years, right? After that, this is as a first one have to make it, and second one is the 1880 Charles Laverin. Who is that? Charles Laverin. He discovered the Plasmodium parasite in the blood of malaria patient. Got it? He collected the blood from the malaria patient. He found the Plasmodium, right? And he is the what from Patrick Manson. Malaria might be transmitted through the mosquito. What is the discovery of Charles Laverin? He found the plasmodium in the blood of the patient, right? Third one, in the 1897, Sir Ronald Ross, right? He identified the oocyst. This is the oocyst. Oocyst in the stomach wall of the female Anopheles. Female Anopheles gut is there, no? In that, he found that oocyst. Oocyst is located like this. This oocyst is Plasmodium oocyst. He found. Then it's confirmed malaria is spread through the mosquito. Right? Later, 1898, G.B. Grossi et al. G.B. Grossi and et al. G.B. Grossi et al. Means what? No, group of scientists, co-scientists. These all described, these along with him, all described that life cycle of Plasmodium vivax in the female Anopheles. Right? They found the life cycle they explained in the Anopheles mosquito. Right? Here in this slide, you learn about the Patrick Manson. His discovery is the mosquito might be cause for the malaria. Charles Laverin contribution of this is the he found the plasmodium in the malaria patient blood, and Sir Ronald Ross identified the oocyst in the stomach wall of the female Anopheles, right? And Grassi et al. They describe the Plasmodium vivax life cycle, right? Female in the female Anopheles mosquito, right? Next, we'll go for the next Plasmodium species. Actually, I told phylum is the protozoa and subphylum is the epicomplexa. Class is the Telosporia. Now, species. Four species are there. First one is the Plasmodium vivax and Plasmodium falciparum, Plasmodium ovale, Plasmodium malariae. These are the four species are there, right? This is the fourth one. Four species which are there in the Plasmodium. They're causing diseases in human. Plasmodium vivax causes the benign tertian, that fever name. Falciparum is the malignant tertian. Ovale causes the mild tertian. And Plasmodium malariae causes the cordon tertian or the cotton malaria. In this, among four, which is the dangerous and dreadful and oftenly causes, this is the malignant tertian. And here is the one specialist there. Those are suffering from the sickle cell anemia. Sickle cell anemia. That people can show the resistance to the disease because of this Plasmodium falciparum or the Plasmodium species lives in the human RBC. Sickle cell person RBC will be as a sickle shape, right? That's why it's not suitable for the growth of this falciparum. so that people can show the resistance, right? And have to remember vivax benign VB, falciparum for the malignant tertian, ovale for the mild tertian. Malaria causes the quadrant malaria. Then here is the, we can see that host, primary host I told, female Anopheles mosquito. Female Anopheles mosquito is the primary host. That's happens the sexual phase. That's why we call as the primary host, right? Means Plasmodium will be undergone the sexual phase in the mosquito, asexual phase in the man. That's why secondary host. Apart from that, reservoir host will be as a monkey. It will be stayed when the both hosts are not available. It will be lives in the monkey, not cause any damage. And which stage is the infectious to the human? That's nothing but sporozoite. What do you call? Sporozoite stage, which is in the sickle shape. This is the causing to the human disease stage. How it will be transmit infected? When mosquito bites the person, infected mosquito bites the person, then that will be called, that mode is called as a inoculation, like a giving injection, like mosquito is sucking the blood, then obviously these sporozoites will be enter into the human blood. That mode is called as a inoculation. Got it? Inoculation, right? These are the very important. Plasmodium vivax causes the which fever? Benign tertian malaria. Falciparum malignant, right? Ovale mild, malaria quadrant, right? These host also remember. Now we'll go for the digenetic parasite. How it is digenetic parasite, right? For the completion of life cycle of the Plasmodium, Plasmodium has to complete life cycle in the two host. That's why called as the digenetic parasite. What do you call? Digenetic parasite. First one is the human phase, second is the mosquito phase. This is the asexual reproduction. We call it as a zygogony. This is the sporogony malaria in the particular mosquito. Where it lives are enter into the liver cell as well as the RBC. But here is the gut or the crop wall. Massive multiplication and symptoms will be appeared here. Here is the gametogony takes place, fertilization takes place in the mosquito, formation of the sporozoites also takes place in the mosquito body. So here liver cell and RBC, the sporozoite entered the particular cycle called as sporogony, right? This is within the cell, liver cell RBC happening. That's why this is called as the intracellular path, intracellular phase. But here is the out of the cell. That's why called as the extracellular. So extracellular mode you can find in the mosquito, and intracellular mode you can find in the human phase, right? So here that will be as a inoculation. Inoculation will be happening. Now, which stage is causes to the human disease causing stage? Nothing but sporozoite. Sporozoites are the disease causing stages for the human being. That structure you have to learn here. This is its shape is the sickle shape. What is the shape? Sickle shape. It has a outer covering for the protection purpose. That is the elastic pellicle, and anterior. This is the anterior part. This is the posterior part. Anterior part have a apical cup, and it has a convoluted tubules are there, secretory granules are there. These release the cytolytic enzymes. Which enzymes? This apical cup, they have a cellular docking. That's nothing but we called as a cytolytic enzymes. These enzymes only essential for the penetrate into the RBC. Got it? When infected mosquito bites the healthy person, infected mosquito, the mosquito have what? Here I have sporozoites. These how many? 20,000 of sporozoites enter into the blood of the human through the inoculation process. Within a half an hour, within a 30 minutes, it will be traveled in the human blood and finally reach the liver cell. Right through the bloodstream, they can opt the liver. Why? Because liver is the good source of glycogen. Right? They enter the first through the bite to the bloodstream, then reach the liver in how many minutes? 30 minutes. Right? These are the sporozoid structure. Right? Now here is the that total hepatic cysogony. Actually, the cysogony in human we are talking about. See here, cysogony. It happening in the human being. It has a one part is the hepatic cysogony, which is happening in the liver cell, and RBC, that is called as erythrocytic cysogony. Right, human face. Now in the hepatic cysogony, that's see this is the liver cells. All are the hepatocytes. Timeline they have given pre-erythrocytic cycle complete in the 8 days, exactly 8 days. Pre-erythrocytic before entering into the RBC. How the entry? Entry is the sporozoite enters in the liver cell. I told you know that is the 30 minutes after inoculation and transform into the trophozoite. Sporozoite transform into the trophozoite. Trophozoite will be changed into schizont. Schizont will be multiply as a schizogony. Then they form as a cryptozoites. Cryptozoites called as a first generation of merozoites. Anyhow, these are all the things happening in the liver cell. Remember, check here first entry, bloodstream, blood to liver cell. This is the liver cell, hepatic cell. Then feeding. That's a feeding what? Trophozoite. Then growth. Nothing but schizont, right? Schizont will be multiply and it will be form as a cryptozoite. That's the multiplication. Then our that liver cell will be ruptured. Number of cryptozoites or the first generation merozoites will be released from the liver cell. Then here is the they will be enter the RBC or else they can attack the fresh liver cell. If they, which one? If they means what cryptozoites may be enter into the RBC. They can start their cycle. We call as a erythrocytic cycle. Or else some will be reach again will choose the liver. We call as a exo-erythrocytic cycle. Now here is the in human phase. Hepatic cysogony liver cells. In the liver, what happening? Liver hepatocytes. Two stages there. That's the erythrocytic cycle and exo-erythrocytic cycle, right? So these which one entered first sporozoite? What will be changed finally? Cryptozoite, also called as a first generation merozoite, right? Here they given as a complete Golgi. Golgi found this one erythrocytic cycle. That's why called as the Golgi cycle. Trophozoite. First one is the trophozoite. Vacuole develops, pushing the nucleus to the periphery. See here, nucleus going to periphery because of the vacuole. This is called as a trophozoite stage. Later, see how it happen. Look like a ring or finger ring. That's a signet ring stage. Then it look like a amoeba, amoeboid stage, and it will be has a in the it happening where no RBC no inside the RBC they multiply and form as a more schizonts and then RBC ruptured and these will be released which we are the while rupturing this hemozoin will be released hemoglobin only will be split into toxic because of the meros these are the merozoites in the previous slide which are reaching the RBC. See, here is the RBC. We are talking about exo erythrocytic cycle. So these merozoites are released from the tropozoites to merozoites are released. Cycle completes how many days? No, 48 hours. Right? Now here is the clinical versus biological timeline. First entry of sporozoite in the blood. Later, there are no symptoms, but second entry of the plasmodium into the blood, like a cryptozoite. At that time, that time span is there now. We call as a preparant period 8 days, right? Entry of sporozoite in the blood. Incubation period 10 to 14 days. The first appearance of the symptom in the human being. The lag between, here is the see here, the lag before the body registers the clinical threat of the parasite exponential growth. See here 2, 4, 6 like that, 14 like that will be keep on double within the 14 days plus, right? Preparant period means for 8 days and incubation period is the 14 days. Here is the second entry of Plasmodium into the blood, the exactly cryptozoites, no symptoms. Later will be have a symptoms, right? Now here benign tertian fever, how it is? Three stages will be there. First one is a cold stage, hot stage, sweating stage. The poor patient will be complaints with the chills, shivering, headache, and giddiness. And hot stage, spinking, high body temperature, rapidly breathing will be there, increase the pulse rate and hotness. Latter profuse the more sweat, followed by the lowering body temperature back to the normal. He felt it is that there is no fever, then it will be start again. Because of the hemozoin pigment and merozoites released into the bloodstream, the spike will be there, hot stays. Right? See here, repeated infections leads to the anemia because of RBC going to split, right? And splenomegaly, spleen going to be enlarged, and severe cases that tropical splenomegaly syndrome, that's abnormal immunity there, no, the spleen will be enlarged because of RBC that will be there, no, related to the RBC. That's the reason it will be shows that, right? And in that next class, we'll talk about the rest of the mosquito phase. I hope you clear. Please utilize this one. Thank you. Thank you so much.